Hello, everyone. Welcome to my classroom. This Eagle and I have been diligently reading your textbook so that you don't have to. So I'm going to highlight for you what you need to know for the AP exam about chapter 6, a tour of the cell. This corresponds in AP to the first two sections of Unit 2. So what you need to know about the organelles of the cell. Let's jump right in. So cell biology is, of course, extremely complicated. And you could get your PhD in any number of these topics. So we're not going to have time to go into all the details, but for a basic introductory college level biology course, there is some things you need to know about the organelles of the cell that goes beyond merely being familiar with it, like knowing that the mitochondria is the powerhouse of the cell. What does that really mean? You need to be able to describe the structure and function of each of these cells types. So first, let's talk about prokaryotic cells. You can think of them as the prototypes. These cells are much more simple than plant and animal cells. They're generally small and rod-shaped. And one of the characteristic ways that we identify a prokaryotic cell is that its DNA is free, meaning that even though usually is maintained in one part of the cell called the nucleoid, is not surrounded or protected by a nuclear membrane as in all other types of cells. So it has free DNA. The DNA can be coiled up in chromatin, but it does have chromosomes just like plants and animals, but it's not protected by nuclear envelope. Prokaryotes have free-floating ribosomes. It does not have any membrane ribosomes because it doesn't have any membrane-bound organelles. Everything is, again, free and floating around. What is it floating around in? All of them are floating around in the cytosol. This is just the inside of the cell that will be unique to the particular prokaryotic cell we talk about. So anything inside the cell other than the nucleoid or the nucleus is considered the cytosol. It's the fluid that everything else is inside of. Another thing is prokaryotics can be autotrophic or heterotrophic. If they're autotrophic, they will perform photosynthesis just like chloroplasts. In that case, they will contain pigments if they're photosynthetic. However, they are not going to have any compartmentalization like we'll see in plants and animals. Like all of these will not be existing. Sorry, not including cell wall. So these and many more organelles will not be there. Lastly, most prokaryotes have a characteristic cell wall, and all living organisms have a cell membrane. This allows them to keep their genetic information, nutrients, and other things needed for homeostasis inside the cell. The exact lipid and protein composition of the cell membrane does vary from plant to animal to protist to fungi to prokaryotes, but all of them have a bilayer plasma membrane. Now looking at eukaryotic cells, and in this class we will spend most of our time talking about plants and animals. Plants and animals have a much larger size. Plants are generally blockish, meaning they're more rectangular, and animals are more spherical. In fact, let me put rectangular. It's like a rectangular prism. Or a block, or like a block. What would be both? Both of them, and again, this is characteristic not just of them, but all eukaryotic cells have a nucleus. This is the first thing we look for when we look at a cell to identify if it's a bacteria or is it something like a protist, an animal, a fungi, or a plant. They have ribosomes, and unlike prokaryotes, they have both free-floating and membrane-bound ribosomes. Both free-floating and membrane-bound. They have cytosol, of course. Again, the exact details of what chemicals, nutrients, organelles will be in that cytosol is different from cell to cell, but they all have that enclosed within the membrane. Both plants and animals have vacuoles, but their vacuoles are very different. Plants have a large central vacuole, which allows it to maintain terminal pressure, and animals have small vacuoles with different functions. Plants have chloroplasts, animals do not have chloroplasts. Both chloroplasts and mitochondria have an additional double membrane within their additional double membrane. They're double membrated organelles. In fact, they're very similar in size and function to prokaryotic cells themselves, which supports endosymbiotic theory, the idea that these are the descendants of prokaryotic cells. For example, chloroplasts, mitochondria, and chloroplasts and mitochondria are small and rod-shaped. They have their own free DNA within their matrix. That's the fluid within the organelles. They have their own ribosomes within the matrix. And matrix is just what we will call the cytosol of these organelles. And so because of that, and because they're the only organelles that have these things, it makes them appear much more like prokaryotic cells, again, supporting the idea that they are of endosymbiosis. They used to be their own organelles. Other organelles like the vacuole or the Raphiar or the Golgi have a single membrane and have no such DNA. So they would not be possible endosymbiosis. Then we have the pathway for protein synthesis. Messenger RNA coded from the DNA in the nucleus sent out to the ER. In the ER, the rough ER synthesizes proteins. The smooth ER does detoxification and synthesizes lipids. So the rough ER will send a protein in a vesicle to the Golgi. In the Golgi, sometimes we call it the Golgi apparatus or the Golgi complex. There's two sides. There's a cis side, which receives the vesicle. It will fuse with the Golgi. It will modify the protein, and it will release it on the other side of the Golgi. That means the Golgi is polar. It has two sides which are different and have different functions. That'll be sent out in a new vesicle. And generally, if it was made in the rough ER, it'll be sent to the plasma membrane, where it will fuse with the plasma membrane and release that protein. Some cells will do this much more often than others. For example, something like the pancreas, it has a lot of ribosomes and a very large Golgi because it's often is making insulin and glycogen a lot, and so it needs more of those. So we can see not only do these organisms have specialized organelles within their cells, they're also specialized with different parts of the cells. As we already talked about, plants and animals have mitochondria. Those perform respiration. Chloroplast is only in a plant because plants are able to do photosynthesis. Animals have lysosomes. Plants generally do not have lysosomes. These allow for recycling of the plant cells. In cells like liver cells, which is doing a lot of detoxification, lysosomes are extremely active. But all animal cells need lysosomes to some degree to break down nutrients, break down dying organelles, and recycle them. Plants have a cell wall. Animals are unique for not having a cell wall. And the way they get around that is plants and bacteria all have a cell wall, and they use that for extra protection. Animal cells are more flexible. That's a structural difference that allows for functional differences. So animal cells are better fit for moving animals. That's why it's much easier for animal to move than plants. So we see again, structure matches function. Outside of plant cells, they're connected to each other. The cytosol of one plant cell is attached to other plant cells through channels called plasmodesmata. And those plasmodesmata is again allows for nutrients and even some proteins or RNA or other things to flow between plant cells. Animal cells also have a matrix which connects them to other cells, and they can have different type of connections similar to plasmodesmata called tight junction, and gap junctions, and however you say that word, I don't know. So it's similar to plasmodesmata, but subtly different. Not all animal cells have these, but something that all animal cells have is something called a extracellular matrix, which allows them to interact and communicate with other cells, made mostly out of the carbohydrate collagen. Okay, that was a lot of information. I encourage you to grab a nice study book, like 5 Steps to a 5. Make sure you look at the pictures of each of these cells, you understand them. But just so you know, there's, again, so much more detail than the ones I went into. But these are the parts of the cell that you absolutely need to know their structure and their function and be able to describe their role in animal cells, in plant cells, and in prokaryotes, how all of these cells are different, and how the presence or absence of certain organelles affects their ability to function. For example, let's do an easy one. Plant cells are able to do photosynthesis because they have a chloroplast. Animal cells are not able to do photosynthesis because they lack a chloroplast. That is a fundamental difference in in plant and animal cells that leads to them having completely different functions. Plants being autotrophic, animals being heterotrophic. Hope that helps you. And let me know if you have any questions in the comment section. Again, lots and lots of details. I tried to explain this as quickly and clearly as possible. Sorry if I didn't do it perfectly, but yeah, let me know if you have any questions. Hope that helps, and good luck studying, everyone. Bye.